Hello everyone, welcome to the Dandelion Biology series. My name is Hyunsung or Steve Go, and today's video will be part 2 for the cell biology unit. To start, let me first introduce one of the ways to divide cells into two big categories, eukaryotes and prokaryotes. Basically, prokaryotes are cells that do not have a nucleus to store their genetic information, which have DNA that are naked or don't have histone proteins. We'll go into more detail for that in a later unit, so do not worry about it for now. Eukaryotes, on the other hand, have a nucleus to store genetic information and have a lot of organelles inside them that serve different purposes. So what makes up prokaryotic and eukaryotic cells? Well, prokaryotic cells have all of these parts in front of you right now. On the right is a diagram of a bacterial cell, which is an example of a prokaryote. First, the nucleoid basically refers to where the genetic information is floating around in the prokaryotic cell. Remember that this is not a nucleus or some designated space. Around the nucleoids are ribosomes, which synthesize proteins from mRNA. The 70S in parentheses over here refers to its size. There are 80S ribosomes, which you will see in eukaryotes, which are bigger than these 70S ribosomes. Plasmids are round strands of DNA that are separate from what we refer to as the chromosomal DNA of the prokaryote. They are used in bacterial conjugation, a form of sexual reproduction in prokaryotes for genetic diversity, in contrast to binary fission, which is the common method of reproduction amongst prokaryotes where they basically clone themselves. This is asexual. Cytoplasm is the thick solution that fills the cell, surrounded by the plasma membrane, which regulates material exchange, which is surrounded by the cell wall, maintaining the shape of the cell and protecting the insides. It is again surrounded by the slime capsule, which protects the cell from physical and chemical threats. Now we move on to eukaryotes, which share some parts with prokaryotes. They have ribosomes, ADS, which are bigger than those of the prokaryotes' 70S ribosomes, and they also have cytoplasm alongside a plasma membrane and a cell wall in case of plants only. The first structure we haven't looked at yet is the nucleus, which stores most of the cell's DNA inside it. I say almost because the mitochondria and chloroplasts do have separate DNA from the rest of the cell. Then we move on from the nucleus to the endoplasmic reticulum, which has two parts. The rough endoplasmic reticulum, which contains ribosomes, they perform the function of synthesizing proteins for use outside of the cell. It's in contrast to the free ribosomes, which produce proteins for use inside of the cell. The second part of the endoplasmic reticulum is the smooth endoplasmic reticulum, which is in charge of synthesizing which is in charge of synthesizing lipids, not proteins. The Golgi body is an organelle that packages proteins to be sent outside of the cell. Lysosomes are basically the digestive system within a cell, in charge of breaking down material or sometimes parts of the cell itself. There are vacuoles present both in plants and animals. These are used for storage of water, salts, or any waste in the cell. The things that are exclusive to plants include the cell wall, as said earlier, and chloroplasts, which perform photosynthesis. Now we shall look into one of the parts of both prokaryotes and eukaryotes, the membrane structure. Throughout history, different theories arose to explain the layer of wrapping around cells. An example of an unaccepted theory is the Davzin-Danieli model, which is nicknamed the sandwich model. Basically, it's a bilayer of phospholipids, we'll get into that later, sandwiched between a layer of proteins. The currently accepted model is the Singer-Nicholson model you see over here, which has a bilayer of phospholipids with proteins and cholesterol. Phospholipids are made up of phosphate heads and fatty acid tails. The phosphate heads are hydrophilic, which means that it has a tendency to mix with water, so it heads outwards. The tails are hydrophobic, which doesn't mix with water, so they face inwards. There are three types of proteins that you can see on the diagram. They are integral, which pass through the phospholipid bilayer, peripheral, which only pop out of one side, and channel proteins, which allow for substances that can't pass through the membrane on their own to pass through. Substances that can pass through are very small, like oxygen or carbon dioxide, or are hydrophobic. Finally, cholesterol binds the phospholipids together, providing structural integrity. Remember that this is not a solid, moving structure. The whole structure is like a fluid with different proteins moving across the membrane. This gives the Singer-Nicholson model its nickname, the fluid mosaic model. That's all from Dandelion today. If you have any questions, please comment down below. And if you would like to stay tuned for further videos, please click subscribe. See you next time.